This
And
conference will now
in the
be
same
recorded.
we found the dioxin accumulation, and it was found to be around 33 ppm in this aged callus. So this tells us that, yeah, the expression of the genes which is there in the mother plant is also there in the callus, and the callus also accumulates the secondary metabolite. In another study, we used Barleria prionitis. This plant's local name is Vajradanti, and it's used in cream cosmetic industry, anti-aging creams. So here also we established the callus cultures. Here we use two explants, leaf and stem, to establish the callus culture, and we compared the secondary metabolite accumulation in mother plant, means the plant from which the explants were derived, and the callus. So this plant has two main active ingredients. First is SME, which is sangiside methyl ester. It's a glycoside with anti-inflammatory and analgesic properties. And the another one is squalene, which is again a triterpene. It is used as adjuvant in cancer therapy, and also it has role in cosmetics and anti-aging creams. So we compared the accumulation of these two secondary metabolites in callus culture and the respective mother plant, and we found that the SME accumulation in the methanol extracts of the leaf-derived callus was around 2.5 times higher than what is there in the mother plant, and in the squalene accumulation in the leaf-derived callus was around 10 times higher than what it actually, it was not even detected in the mother plant. If you can see the data in this table, this is the leaf of the mother plant, and this is the callus, leaf derived callus, and so there was no detection in the mother plant leaf, but in the leaf derived callus we got this amount of squalene accumulation. So the conclusions of this study were that bioactive compounds were present in the callus culture, both leaf and the stem derived, and these cultures showed higher antioxidant activity and bioaccumulation over mother plant. If you compare the leaf derived callus and the stem, then the leaf derived callus outperformed over the other one, and the methanol extracts of leaf derived callus showed a 10-fold increase in the squalene content and 2.5-fold in the shangiside methyl ester. Subsequently, we tried to isolate the squalene from the callus culture, and we purified it through flash chromatography, characterized it through spectroscopic elucidation and FTIR, and we confirmed that the metabolite isolated is callus culture, is squalene. The next area where we worked is using elicitation as a strategy to enhance the secondary metabolite production. Now, what's the mechanism? Here, the elicitors, which can be biotic or can be of biotic origin or abiotic origin, they are added into the culture medium, and post that what happens, these elicitors are taken up by the receptors present on the plasma membrane and gets into the cell and triggers signal transduction cascades, activates the second messengers, they also come into play, and ultimately what they do is increase the expression of the genes which encode for the enzymes of the metabolic pathway, and that way the metabolic pathway is expedite and we see the enhanced production of secondary metabolites. So here for elicitation we studied phenolic compounds, how they can be elicited in Ruta gravulens. Okay, it's again a medicinal plant, and its active ingredient is rutin, which is flavonoid derived from quercetin, and the whole plant is valued for phenolics and flavonoid compounds. So here what we did, we used chemical elicitors the hormonal elicitors, that is methyl jasmonate and abscisic acid, to see if these can increase the accumulation of phenolics and flavonoids in the plant. At the same time, if they can increase the antioxidant activity of the plant. So we established shoot cultures of ruta using internode explant, and then to the medium we added the elicitors separately, methyl jasmonate and abscisic acid, and we harvested the cultures at different durations: 0 day, 5th day, 10th day, and 15th day post elicitation, determined the growth and antioxidant potential of these cultures. So what we observed was that the shoots which were treated with methyl jasmonate for 5 days, okay, they showed the presence of all the phenolics which were studied except the rutin, okay, compared to the control plant. Okay, this is the control plant. And if we look at the rutin concentration, then abscisic acid after 15 days post elicitation showed accumulation of 123.8 milligram per gram routine compared to the control, which was 34.5. So around 2.5 fold increase. No, this is not 2.5. This was again around 3.5 fold increase in the routine concentration was noted. And the interesting thing is there was no compromise on the growth of the shoots. So if you can see abscisic acid, this is the growth obtained. So actually both methyl jasmonate and abscisic acid could increase the metabolite production without compromising on the biomass growth. And out of the two chemical elicitors, methyl jasmonate was the one which showed the presence of all the phenolics, and abscisic acid is the one which shows the maximum accumulation of rutin, which is the main phenolic, main compound for which this plant is valued. And we also found that the antioxidant activity of found that total phenolic content and total flavonoid content was also improved when the cultures were treated with abscisic acid and methyl jasmonate, apart from the individual phenolics which we just discussed. Another application of plant tissue culture on which we worked was to use these cultures as a model system to study abiotic stress response, particularly the salinity stress response, and again, and using these systems to test the mitigation strategies. So soil salinity is one of the major abiotic stresses which hampers the crop growth, development, and productivity. And so creating better salinity crop is a critical step for viable food production. And we tested the potential of plant tissue culture in screening for stress tolerance, which is a well established system. The advantage of using in vitro system is that the response being studied is in controlled conditions, independent of the environmental cues which are operating at the field level. So here we worked on allium cepa. We studied the salinity-induced modulations in growth and biochemical traits of allium cepa in establishes callus cultures using the allium bulb as an explant. And we found that the callus could grow at 100 millimole of NaCl concentration, and subsequently it declined with the increase in the concentration. And then we tried to study what's the mechanism which allowed the callus to survive at this temperature, at this salinity condition. And we found that it's the proline accumulation was higher, and so we can say that it's the osmotic adjustment which is behind the ability of the callus to survive on 200 millimoles of concentration. And if we look at the antioxidant activity, then peroxidase activity was found to be highest at 200 millimoles. So that could be the reason for the callus surviving at this concentration, although the growth declined compared to the lower concentration, but it still survived, it didn't die, and that could be the reason. That was a very small study we did, and further studies are here needed to find out the mechanism underlying the ability of these callus cultures to survive. Another interesting study we did was stress mitigation. So use of chemical ameliorating agents like plant growth regulators is the most widely adopted practice for management of crop production under salinity. And we tried this strategy in Gingiber officinale. So ginger is valued as a spice as well as a herbal plant, and it has high economic importance. The valuable part for ginger is rhizome, and that's the part through which it is propagated. Ginger productivity is known to be adversely affected by soil salinity and thereby affecting ginger industry. So we tested the use of salicylic acid to attenuate the symptoms of salt stress in ginger plant. And for that purpose, we established the cultures using rhizome as an explant. And the experimental material used was the hardened plants. These are the hardened plants. You can see these are in the artificial soil. Okay, so after the root induction, the plants are transferred into this artificial soil called soil right for hardening. So at this stage, we applied stress to the plants, and at the same time, the ameliorating agent, that is salicylic acid, to the plant, and we recorded the observations on growth and biochemical parameters. We used two modes of application of salicylic acid. One is root application and another is foliar spray. And as you can see in the figure, the foliar spray showed better growth, more number of roots compared to the root application. So this is the graphical abstract we presented in this paper. So that makes clear. This picture is when the plant was exposed to 150 millimole of NaCl stress. And when we investigated the biochemical and growth parameters, there was a decline in biomass, chlorophyll content, carotenoid content, antioxidant activity, and like that. And there was enhanced iron toxicity, chlorophyllase activity, superoxide radical generation. And when the salt stress was applied along with the ameliorating agent, that is salicylic acid, its foliar spray, then the presence of salicylic acid reverse these parameters. So higher biomass, chlorophyll content, around threefold higher increase in shoot number and fresh weight was recorded. The chlorophyll content increased by 41%, and the carotenoid content increased by 36%, and there was reduced chlorophyllase activity. And then iron toxicity was also reduced. It was reversed. It was as indicated by the decline in the concentrations of sodium and chloride with a concomitant rise in the potassium ions. And so we can conclude that acid treatment, the salicylic acid treatment, could be suggested as a feasible approach to produce salt adapted ginger plants in vitro. And such plants can then further be tested under field conditions to see their performance. Another application of plant tissue culture is to use it as a tool for clonal propagation and germplasm conservation. For this purpose, we worked on Chlorophytum borevillianum, which is commonly called as Safed Musli. It's a rare medicinal herb. So we optimize the factors influencing shoot multiplication, like the concentration of the agar we optimized and found that 0.4% is showing maximum growth. Then the method of subculture was tested. When you subculture, you place, either you place the initial culture directly onto the fresh medium, or you divide the initial culture into small parts and place, and what's the difference. And we found that this is the method, best method to get large number of shoots, when you divide the culture, initial culture, into small clusters and then place it onto the fresh medium. Similarly, we tested the influence of different vessel size, because different vessel size with different stoppers, and that also showed some difference in the growth. Like you can see the bottle with a vented stopper, this is a vented stopper, and it showed the maximum growth, because in vented stopper the ethylene accumulates in the vessel, and through the vent it it's released, and that and so the inhibitory effect of the ethylene is reduced, and that could be the reason for higher growth, is what we uh, explained. Then we also tested the effect of carbon dioxide environment on the growth, replacing sucrose, and we found that if the a controlled carbon dioxide environment can be a better alternative over adding sucrose into the medium, and that resulted in improved growth and showed proliferation in chlorophytum botulinum. Then we worked on Feronia limonia, which is again a medicinal plant, and we established its adventitious shoot cultures for clonal multiplication. Then, in basic research studies, we studied the stomatal characteristics of Wrightia tomentosa, which is again a medicinal plant, and we found here that the stomatas under culture conditions, they are abnormal. Their guard cells are wide open, and so because of this abnormality, we need to subject the plants to hardening and acclimatization, which is an established thing, but we tested what's happening to the stomatas under in vitro condition. Then we worked on Helicteris isora, where we tested the effect of light and temperature on secondary metabolite accumulation in callus culture. And in another study in tobacco, we tested the effect of gelling agents and carbon source. So these are the few plant species where we optimize the rate of shoot multiplication in order to obtain plants clonal propagation. So this was the first aspect. Second is when we produce the plants through tissue culture, the most important thing is to test whether the plants are genetically identical to the mother plant or not. Because when we say clonal propagation, this is what it means, that daughter plants have to be identical to the mother plant. So that can be tested using various molecular and cytological tools. And in our study, we use RFPD as a marker to test the genetic fidelity to tightness of the tissue culture raised plants, and we found that to be identical. Then for conservation, we also tried one more method, and that is encapsulating the shoot buds in sodium alginate bead. This is how the bead looks. You can encapsulate it, and storing these beads under different conditions at different temperatures, and then testing the viability of these beads at different durations to obtain the plant. So that was another study which we published in European Journal of Horticultural Science. So apart from molecular markers, the cytological markers can also be used for testing the fidelity. And here in this study, we screened the microclones of chlorophytum for their stability at different culture age. Means how old is the culture? 1 month old culture, 2 months, like that. We did it for 2.5 years old cultures of 3 different accessions of chlorophytum borevillinum, and we found that the cultures showed genetic fidelity with the mother plants. They were absolutely identical even after maintaining the cultures, multiplying them for 2.5 years. We also studied the cytological characteristics and found that there was no change in the ploidy level and the karyotypes of the tissue culture plants over the mother plants. So that was all the work which we have done in different applications of plant tissue culture. Now coming to the emerging trends in biotechnology, what we have: precision breeding with CRISPR-Cas9. So it's a precision gene editing tool where we can make site-directed changes in the genome, overcoming the limitation of the agrobacterium-mediated transformation. Then synthetic biology is another area where engineering the metabolic pathways of the secondary metabolites to produce the metabolites in masses. Then multi-omics approach, integrating your genomics, transcriptomics, and proteomics for improving the crop and understanding their interactions. Then digital phenotyping and AI is another emerging trend where using sensors and imaging devices to collect the data, the phenotypic data, the mass morphological data of the plants grown under different conditions, stress in the stress condition, under different nutrient levels, and then predicting the subsequent effects. If the plants are exposed to stress, what will be the effect? So based on that data, the predictions can be made. Then use of biofertilizers and biostimulants to increase the growth of plants under field conditions. Molecular farming is another emerging area where using tissue culture system as bioreactors for large scale production of useful compounds. Till now, what we were talking about use of tissue culture for clonal propagation, for mass propagation of plants. But in molecular farming, we are producing tissue culture plants, various tissue culture systems. But the purpose is not to get the plant. The purpose is to produce the secondary metabolites, okay, or to enhance the secondary metabolites by recombinant engineering. And that is what is the application about molecular farming, especially the pharmaceutical compounds like proteins and antibodies, vaccines. They are produced in transgenic plants through molecular farming. Then vertical farming is another emerging area where tissue culture plants are grown in hydroponics system. So that is not very much tapped, but it's an emerging area. And then the last one is producing secondary metabolites other than the pharmaceuticals, like your bioplastics, then green energy, such things, producing using plant biotechnology methods. So I will end this talk with an inspiring note that plant biotechnology offers the promise of resilience, innovation, and sustainability. And it empowers us to unlock the full potential of plants, not just as source of food, but also as solution for health, energy, and environmental restoration. So thank you very much. The floor is open to questions, if any.
Thank you. Thank you so much, Dr. Joshi, for such an insightful presentation. And yeah, it was really amazing to know how salicylic application can significantly improve the growth, photosynthetic pigments, and antioxidative enzyme activities and imbalances in ginger plant sets, which are under induced like salt stress or oxidative damages. So this approach could actually help, produce salt-adapted ginger plantlets suitable for cultivation in saline soils or, plants or crops which are under anthropogenic stress. So yeah, thank you so much. Um, we now have time for a few questions from our audiences, and please type your questions into the chat box, and we will address them one by one.
In case you don't have any questions, you can reach out to me on my email. or you can reach out to Niharika, and
Yeah,
I'll be happy to answer your queries.
yeah, yeah. Um, so any questions from anyone right now, if you're having?
Yeah, I can see one questions. Let me take. Niharika, you are going to take, or shall I take the questions?
Yeah, yeah, you can take. It's not a problem.
Okay. So
So
one
I guess
is
this one, right?
Aman.
Mhm.
Aman is saying, is it possible to work under you and also get the session recording? Yeah, for that, Aman, you can reach out to me on my email. And Harshini is asking, ma'am, can you please tell me the species that you have worked? So, which plant you're talking about? I have named, I think, all the plant species on which I work. Can you specify your question, Harshini? Like I worked on Chlorophytum borevillinum, then Ginger officinalis, Ruta graveolens, then Costus speciosus, then Ferona limonia, and Wrightia tomentosa. So, what exactly you are asking, if you can tell me? Aman, one more question is, which plant can be adopted for toxic any model? Yeah, Aman. So, you can use
Plant cost of
Harsh, a sound is
tobacco.
coming from. Hmm.
Tobacco can be a very good model system to test the toxicity. Okay,
ma'am.
Vivek, question is I would like to answer. Yes, Niharika.
No, it's not me. I guess it's Harshini.
Yeah, Harshini, tell me.
Ma'am, I want to know about the costs which you have told.
Okay, Costus speciosus.
Yes,
The
ma'am.
species speciosus. Okay, I would like to take Vivek's question, ma'am. What is the role of tissue culture in stress mitigation? So tissue culture can be used as a model system to test the influence of various emulating agents in mitigating the stress. And once it is tested and we get the positive response in the tissue culture, we can extrapolate the responses obtained in tissue culture to the field level. But yeah, we have to validate those responses. So it is the first step, we can say, first step, first hint we get whether the emulating agent which we are using is going to mitigate the stress or not. So instead of testing in the field, we can test first at the in vitro level, and then we can extrapolate it to the field level. What else? Yeah, Anant is asking how far your research turned into a marketable one and difficulty phase. Anant, we have yet to reach to that stage of marketing the things and commercializing the research. Poonam is asking, can we
Intelligent.
use endophytes from Barlaria prionitis using it in tissue culture? Yes, of course. There are a lot of reports where endophytes isolated from the tissue culture plants are used for improving the growth and rooting and hardening of the tissue culture plants. It's a well known strategy, and the endophytes. So using endophytes over the plant growth regulator has also been found to be very beneficial. So that way, cost cutting is also done. You don't need to add IA or any growth regulators at the endophytes. But then again, you can try. There are a lot of success reports of using endophytes for improved growth. So of course, you can try in apple rooting. Harshini is
Attending.
asking, is there any heterologous expression in Costus speciosus? Heterologous expression, there must be, because it is a well studied medicinal plant, but right now I'm not sure what genes have been expressed in costas, but definitely for improving the metabolite content, the metabolic engineering is done in costas species. But what exactly the genes were transferred, we need to find out actually. Hope I have taken
Any more
all the
questions?
questions. Niharika, if you can help me
Yeah,
and if it makes
yeah, yeah,
any.
yeah. Yeah, I think for now we can actually we're actually currently at the end of the meeting.
Okay.
So if there are any more questions, so as ma'am said that you can actually reach ma'am through email or you can reach us and we will try to connect you with ma'am. So yeah, Anand is asking for your mail ID, ma'am.
uh, can I type in this? Okay, I'll
Yes,
type in
you can type in the
just a minute.
chat box, ma'am.
Everybody. So here you go, Anand, and you can reach out to me and we can answer. Just a minute. There is but one. There is a mistake, I think. This is the correct one. Everybody, please note.
I'm just pinging this message so that it will be helpful for others, I guess. So yeah, as we conclude today's session, I want to extend a heartfelt thanks to Dr. Joshi for sharing her expertise and to all of you for your active participation. It was such a nice question and answer session. It was such an interactive session. So thank you everyone. We appreciate you being here. And please, I'm just sharing one link, one feedback link in this chat box. Please go through it and ensure to fill out the feedback form so that we can improve our future webinars and provide the best experience for you all. Please make sure to fill up this form before leaving this meeting so that we can actually provide you, because without this filling up this feedback form, we can't provide you the certificate. So please ensure to fill it up before you leave this meeting. And we hope this webinar has provided you with valuable insights to guide you your career journey. Please stay connected with Biosetup Life Sciences for more such enriching opportunities. Have a great day ahead. And thank you, Dr. Joshi, once again
Thank you
for
very much.
your time and support. So,
Thank you, everyone. Thank you,
and
Niharika, for giving me this opportunity, and hope to do this more in future. Thank you.
yes, ma'am, sure. Sure. So please, requesting everyone to fill up this form before you leave this meeting. So, once you're done with the form, you can leave the meeting.